Welcome everybody. I'm happy to start the 15th edition of the 15th webinar of our online edition of the protein electrostatics. The presenter of today is Thomas Simonson from the Ecole Polytechnique de Paris. Before introducing Thomas to all the participants, I would like to add some announcement. So the next week, the speaker will be Marilyn Gunner, and afterwards we will have a short roundtable. I would like, so since I guess the month of May will be the last one, probably, or maybe the beginning of June, based on the submission, the webinar series will be finishing. So I think at some of the next presentation, it would be nice to have some short roundtable on some topic. And I think we will start next week with the roundtable. So the topic could be something around at which point are we in computationally estimating the charge of a biomolecule. That it has been a topic particularly recurring in the past meetings. So coming back to the speaker of today, it's a pleasure for me to have Thomas Simonson today. He is an engineer by background, then he has got his PhD in 1985 at the University of Paris-Sud-Orsay in condensed matter physics. Then she moved to the synchrotron lab of the CNRS. Then he was associate research scientist at Yale University at the biochemistry department. And eventually he came back to the CNRS in 1992 at Strasbourg. And now he is director of the Laboratoire de Biologie Structurale de la Cellule in CNRS in France. And the director, and he is also the head of biocomputing team. Among the other activities, he is the developer of the Proteus protein design software. The title of his presentation of today will be Computational Enzyme Design without Rosetta. Welcome.
Thank you,
Thomas.
Walter. Thank you very much. I want to join all the speakers in kind of saluting this wonderful effort Walter makes every week to bring us together. I mean, it's been scientifically a lot of fun and very useful, and also really nice to see, if not exactly friendly faces, at least friendly names in our participant list. So I think, yeah, I'm set to share my screen. And okay, this should be visible. So how to design enzymes without Rosetta, is that even possible? So no disrespect to the Rosetta community. Rosetta is a very big feature in the protein design landscape. But a number of years ago, we set out to build up our own tools using our own expertise in molecular simulations. And so before I get to enzymes, I need to give you a certain amount of background, especially background on the methodology we use. Of course, all of us here are interested in the folding problem, right? So I give you a sequence and you give me a fold. The inverse problem is also very interesting. You give me a fold and I give you a sequence. So that's one of the things, one of the problems that protein design addresses is this inverse folding problem. Of course, the size of the space of solutions is enormous. And the set of natural solutions is also quite big. For example, for the PDZ fold, there are over 10,000 natural homologs in UniProt. These are, a few of them are shown here in the form of a logo, a sequence logo for the hydrophobic core. So this is one of the things protein design does. It searches sequence and conformational space for preferred sequences according to some criteria. And the search could be done with Monte Carlo, could be done with some exact method like dead-end elimination, or could even be done with molecular dynamics. But in all cases, the idea is to propose random mutations and then apply some kind of selective pressure. So if you want to design for protein stability, say, you would start with a particular fold and challenge it with a lot of random sequences and basically filter out the ones that stabilize the fold to a sufficient extent. So for stability design, you're going to need a structural model of the folded and unfolded states and a free energy function, which could be completely knowledge-based or much more physics-based or somewhere in between, which is actually the case of Rosetta. We think what is pretty much a physics-based approach for two reasons. One is the energy function. So we use molecular mechanics for the solute, generalized Born for the solute. We have a nonpolar solvation term. And our favorite one is the Lazardis-Karplus functional form, which we've parameterized to be a nonpolar term. We typically have one or a few backbone conformations and then a discrete library of rotamers for side chains and ligands. And we explore this space using replica exchange Monte Carlo, which is a lot more efficient than simulated annealing, say, and gives you much better control. And that's the second aspect that is really physics-based, the fact that we make a significant effort to do Boltzmann sampling, to sample a Boltzmann ensemble. So that's actually quite simple in a lot of situations, but there are other situations where we really need to develop quite sophisticated Monte Carlo algorithms to accomplish Boltzmann sampling. And of course, Boltzmann statistics is the statistics that are obeyed by actual experimental systems. So that's kind of a nice thing to try to replicate. We have a review article a few months ago that covers a lot of this methodology. But before I go on, I want to say a little bit more about the statistical ensemble, because you might think Boltzmann sampling in a space of protein sequences. I mean, how is that possible? What does it even mean? It's not like the protein sequence can fluctuate, can have thermal fluctuations, right? So in the protein design field, people don't really worry about that, but I just want to split a theoretical hair, if you'll give me a moment here. It turns out that when we make a Monte Carlo move that changes the sequence, so we might mutate sequence S into sequence S prime by changing a particular residue from black to gray, what happens is we actually do the reverse mutation in the unfolded state. So the unfolded protein is going to undergo the S prime to S change. And of course, when we do this double mutation, effectively we're unfolding the sequence S and refolding the sequence S prime. So we're actually making a move in a conformational space. And so ordinary statistical mechanics are going to apply. And if you think about it a little bit, it turns out that the Boltzmann statistics for this ensemble actually mimics an equimolar collection of sequences which are at equilibrium and which are distributed between the folded and unfolded states according to Boltzmann statistics. So that's really the kind of, basically the same as an experimental combinatorial library of sequences, which is nice. We built up software which in its current form is reasonably user-friendly, it's well documented, has a pretty solid set of tutorials. So our program is called Proteus. It descends from Explore by Axel Brunger, my boss in an earlier life. And of course, Explore descends from Charm, which was developed by the Karplus group. Now, before I talk about enzyme design, I want to talk about a benchmark calculation, which is an important proof of principle and a classic benchmark for protein design. And that is the complete redesign of a protein. So in this case, 2 PDZ domains. Now, redesign in our hands means simply running a billion steps of replicate exchange Monte Carlo and extracting the low energy sequences, the ones that have the highest populations. And in these simulations, we have about 3/4 of the sequence is allowed to mutate freely. In other words, we maintain the native residues. Some of the protein keeps its native character because these are the residues here in yellow that are going to bind the natural peptide ligand. We want to be able to assay experimentally peptide binding for our designed sequences. So when we do this, we get design sequences that resemble natural sequences very nicely. So here you have our logo for natural PDC sequences from PFAM, and these are logos from simulations of our 2 different PDC domains, which have slightly different backbone structures. And down here we have similarity scores versus the PFAM PDC domains. And if we went through this in detail, we have a horizontal similarity axis, and then there's a histogram axis. It turns out that for one of our domains, we get nice similarity scores that are a little below the Rosetta results, in our hands at least. And for the other one, we get results that are a little bit above the Rosetta scores. So we get design sequences that resemble natural sequences. Now, we were able to convince experimental colleagues to test some of our sequences, but just a handful. They were really only able to test a handful. Turns out that 3 were tested, and all 3, for all 3, we have good evidence they adopt the correct fold. So they have native-like circular dichroism spectra. They have 1D NMR spectra that are structured, well-structured, similar to the native protein, and very different from failed designs, which have broadened peaks. And for 2 of them, we have an upshift of the thermal unfolding in the presence of the native peptide ligand. So complete protein design has been going on for many, many years, but it turns out this is actually the first example with a physics-based energy. There are very few that have not been done with Rosetta, and the ones that have not been done with Rosetta were distinctly knowledge-based energy functions. So this is the first example with a physics-based energy. Now, for this audience, I do have a few slides that are specifically electrostatic, and this is one of them. What I want to say here is that from our experience, the quality of the solvent electrostatics is important for these results. We believe that the relatively high quality GB model has actually made a difference here. Now, we all know that continuum solvent is a many body solvent. It's not pairwise additive. The field on a particular atom depends on how buried it is, which depends on the shape of the whole protein. So it's a many body potential, and in the GB model, this is of course captured by the solvation radio. But in protein design, it's very convenient to have a pairwise additive energy, because then you pre-compute interactions and put them in a lookup table, and the Monte Carlo was very, very fast. So in the field, everybody outside our group, or other than close collaborators, introduces an average effective dielectric boundary, which kills the many-body property. And now you can have nice lookup tables. It turns out that you can recover the pairwise complexity with a double lookup procedure, which is kind of complicated. I just want to allude to it and go through it very fast. The point is, for each residue pair in our protein, we're going to compute the interaction energy in the deeply buried limit and the highly exposed limit, and also for some intermediate values. And then we parameterize this interaction as a function of burial as a power series. Then later on, and we can basically put these coefficients into a lookup table. And later on during Monte Carlo, we can track the solvation radii, which are pairwise additive, and then recover the interaction energy. So I'm going through this very fast. It's really just to allude to it. The point is we maintain the many-body character of the solvent, and that's very unusual in the field, and we think it makes a difference. So this brings me to computational enzyme design. In our hands, we're actually interested in redesign. We've always targeted the redesign of existing enzymes. So what we want to do is choose a few amino acids in the active site and explore mutations in search of some new activity. So perhaps a new substrate undergoing a similar reaction or a related reaction. So an enzyme redesign. And the criteria we would like to target are actually multiple. Of course, we want to maintain the stability of the protein. We need to bind the substrate. And we also want a reasonable reaction barrier, right? So targeting substrate binding, say, is actually quite difficult because it's a competition between an unbound and a bound state. But this collection of criteria can be made a little more precise and explicit if we go sort of to enzyme textbook, right? So we go back to the kind of textbook ideas we teach to our biochemistry students, and we have things like this free energy diagram where there's a progress variable horizontally, and then the free energy goes vertically. So as usual, we have over here the enzyme and the substrate, which are separated. Here we have the bound complex, the Michaelis-Menten complex. Now we have the chemical reaction step, and then we go over to the separate enzyme and product, right? And we all know there's this free energy sum rule, classic sum rule, which says that the lowering of the catalytic barrier relative to the uncatalyzed reaction is the same thing as the lowering of the transition state binding relative to the ground state binding, right? That's what we tell our students. So we could conceivably target, as a design target, the specificity of the transition state binding. That would be a nice, simple, rigorous target. Turns out we don't really care too much about having a super duper catalyst. It's more important to us to have a sufficient turnover rate. And so we go a little further, we look at the Michaelis-Menten reaction rate, and of course, the second order rate constant is the ratio Kcat over Km, which enzymologists apparently call the catalytic efficiency. And the log of the catalytic efficiency is actually the binding free energy of the transition state. Okay, so we're going to be more interested in designing for catalytic efficiency and also for the specificity of the catalytic efficiency. That is, we might want to increase the efficiency for our new substrate, but we're also happy if we can decrease the efficiency for the old one. So improve the competition between the two. So in our applications, which I'm going to describe in a moment, we will be targeting substrate binding sort of early in our experience, and then we moved on to catalytic efficiency and its specificity. So again, it's quite tricky to design for substrate binding or transition state binding. And the reason is there's a competition between apo and holo states. That means we need positive design for the holo state and negative design for the apo state. Okay, and that's actually quite tricky to achieve. So after a lot of trial and error, we actually discovered a way to do that. And the way to do that is to apply a variant of Wang-Landau adaptive Monte Carlo. So having figured that out, we then went back and checked the literature, and we found that, unfortunately for us, this had already been discovered by a Swedish physicist. Okay, but we were still, quite happy to have this method now we could use. And this is really a rigorous method to sample by affinity. So there are two steps. The first step is to adaptively flatten the free energy landscape of the apo state, the landscape in sequence space. So we're basically going to flatten the free energy of the sequences and make them all roughly equivalent. Then we add the ligand. We simulate the holostate. Okay, sorry. So to adaptively flatten the landscape, we achieve this with a bias function, an additional energy which can be considered a bias function. Okay, so when we add the ligand to form the holostate, we simulate the state, including the bias. And as you can see, or you will see in a moment, the bias actually subtracts out the apo free energy. Simply because if the bias flattens the apo free energy, it means that it's the opposite of the apo free energy. So including it here automatically subtracts out the apo free energy. Now this is an example now where we designed an enzyme by allowing 3 active site positions to vary. Okay, and they can be distinguished here in this logo. So in the apo state, if we run a long Monte Carlo simulation without doing anything special, we have certain preferred types at each position, which can be seen in the logo. And for example, the native types are L, A, and I. And there are other types that show up in the simulation. Now we run a Monte Carlo phase where we adaptively build up this bias in a kind of unsupervised learning step. We build up this bias until the landscape is sufficiently flat. And you can see now that it really is quite flat because each of the positions is now sampling all the amino acid types with comparable probabilities. So we have indeed achieved a bias function that is close to the negative of the apo free energy. Now we add the ligand, and these are the sequences we sample. And again, the native types are L, A, and I. And because of the nature of the bias function, it turns out the sequences are populated now according to their binding free energies. So that's actually quite a remarkable achievement with this algorithm. So how does the adaptation work? Well, I think metadynamics was probably invented by or inspired by Wang Landau Monte Carlo, which is older. But we didn't really know about Wang Landau Monte Carlo. We knew about metadynamics. So we were inspired by metadynamics. It's kind of a circular process here. So here we have an example where there's a sequence axis and then a free energy axis. So these are the sequence free energies, and the sequences here form a basin. They form a single well. So early in our simulation, we're probably going to be, moving around within this well. And every so often we check what is the sequence we're visiting, and we add a penalty, an energy penalty to that sequence, a little red blob here. And a little bit later, the simulation will have moved over a little bit to another sequence nearby. We'll add a little pink blob. And over time, the whole bottom of the well starts to turn pink. And, as we continue, the whole well fills up with pink or red. And eventually the well has been flattened. Okay. And when the well has been flattened, it means that this red object really is the opposite of the gray object. And so the bias is very close to the negative free energy. We don't need a perfect convergence to the negative free energy. It just has to be reasonably close. So in the second step, when we simulate the bound state, including this bias function, now we have a Monte Carlo simulation, which is controlled by literally by the binding free energy. And so tight binders are going to be exponentially enriched during the simulation. There are many ways to use this idea. Okay, what I just described was to flatten the apostate and then sample the holostate. And that means we're actually designing for affinity. But we could also flatten the system in the presence of a ligand A and then sample in the presence of a ligand B, which means we're targeting the specificity, the B versus A specificity. Okay, so we can target that. We can also flatten the complex with a substrate and then sample the complex with the activated substrate, the transition state. In that case, we're literally designing for catalytic power. And we could also flatten the apostate and then sample the complex with the transition state. And if we do that, now we're directly targeting the transition state binding free energy or catalytic efficiency. Okay. And of course, we use a molecular mechanics model of the transition state, which we have to build up ahead of time. So this brings me to our the application I want to talk about. And the application, or the small set of applications, have to do with designing amino acyl tRNA synthetases in view of expanding the genetic code. And I think most of you know amino acyl tRNA synthetases attach a specific amino acid to a specific tRNA, which is characterized by a specific anticodon at the other extremity. And so the synthetase selects the amino acid and the tRNA, including its anticodon. The synthetase is actually the one that establishes the genetic code. It establishes the correspondence between the amino acid and the anticodon. And by engineering them, one can expand the genetic code to include non-natural amino acids. And this was first done by Peter Schultz and a few other people in the very late 90s. And these people engineered a couple of different synthetases to accept non-natural amino acids as substrates, as preferred substrates. And a couple of the early ones are shown here. These are tyrosine analogs that were worked with the tyrosyl tRNA synthetase. So 20 years later, there are over 200 non-canonical amino acids have been added to the genetic code, largely by engineering a handful of synthetases. The first application where we succeeded was the redesign of the tyrosyl enzyme to preferentially bind D-tyrosine instead of L-tyrosine, to bind it and transform it. Of course, D-tyrosine has a non-standard backbone, and to introduce D amino acids into the genetic code in a controlled way, genetically encoded, I mean, this would be extremely useful for protein engineering, for example, for engineering the active sites of certain enzymes where a D amino acid can do the job and perhaps an L amino acid cannot. So we obtained a design mutant that had an inverted specificity, and the actual change in the D versus L ratio was about a factor of 800. Unfortunately, the activity was a bit low, but the specificity inversion was very, very successful. And this was done by allowing just 4 positions in the active site to mutate, and using the kind of old method, which most people are still using, where the mutants are actually selected based on the total protein ligand energy, not the binding affinity, let alone the catalytic efficiency. But the old method did allow us to redesign the synthetase for a non-standard backbone. So now we're working more actively on the methionine enzyme, and which attaches methionine to its associated tRNA, and this has been used for to engineer activity for a number of non-natural amino acids, including azidonorleucine, homocysteine, and a few others. And the ones I'll be looking at are methionine, azidonorleucine, and beta-methionine. I'll come to that in a moment. So now we have this new methodology at our disposal, and hopefully that's going to give us a better handle on the enzyme design. So we tested redesign for the natural substrate, methionine, redesign for azidonorleucine, which has already been sort of covered experimentally, and finally, the real application we're interested in is beta-methionine, which is another non-standard backbone. So our first test was a retrospective test. We tried to recover the experimental results for azidonorleucine. So azidonorleucine looks a lot like methionine. Of course, there's no sulfur, and we have this N3 group attached to the extremity. And this is something that has an infrared signature, can be used for protein labeling and cell localization within the cell of certain objects. So the methionine synthetases were engineered by Terrell at Caltech in 2009 by directed evolution, and they came up with 21 active variants, and actually 6 of them were especially active. They had 3 positions they targeted in the active site close to the ANL substrate. So we basically recapitulated this study using the same 3 positions. Our design criteria was it was not yet catalytic efficiency, it was substrate binding. The simulation sampled about 1957 variants. And among the top variants for the binding affinity, there were a number of experimental variants. So 5 of the 6 top experimental variants were among the top 100 Proteus variants based on binding affinity, which is nice, but perhaps not tremendously impressive. What is a bit nicer is that for the specificity of binding, 2 of the top 2 experimental variants were among our top 10 predictions. So if we were to give, if we did this in a blind way, we might give to experimentalists the top 10 affinity variants and the top 10 specificity variants. We would have 2 actives among those 20. So that's a 10% success rate, which is actually very, very reasonable in the enzyme design field. Okay, so this is a retroactive study. Now we move on to a fully blind study. And now we're going to redesign the enzyme for its own substrate. So we're going to look for new variants of the enzyme that are active for MET. And now we moved on a little bit with our kind of building up experience. Now we're designing for the catalytic efficiency directly and the transition state binding. And we again have 3 positions we vary around the substrate side chain primarily. So this was a fully blind study. We picked out our top 10 predictions and showed them to our experimental collaborators. And they picked out 17 that were a little bit easier to produce as a group. They do PCR mutagenesis, so some sets of variants can be done in a more efficiently than others. They picked out 17 of the top 40, and all 17 were active. So that was a nice result. It turns out the computed affinities were also, these are the transition state affinities, were also in quite reasonable agreement with experiment. Now I'm going to cheat a little bit and show you the computed affinities for not exactly the 17 variants, but for the point mutations we can build up from these 17 variants. Okay. From our 17 variants, we can build up about 30 point mutations, which are of course not independent, but which are shown here relative to the wild type. So I'm showing the catalytic efficiency, on a log scale in KT units, right? So this is the binding free energy of the transition state, and these are values always relative to the wild type. So there's a ratio, there's a dimensionless ratio here in the log. The wild type is shown here at the origin, and you can see that our variants are all a little poorer than the wild type. Some of them are very close to the wild type value. This is the experimental axis here, computational axis here. Perfect agreement is on the diagonal, and we have 1 kcal above and below here. There is one large error which is not on the plot. Okay, so I've left out one large error. Other than that, there are not really any large errors. a bunch of variants are right up close to the wild type. And then the worst variants are about a 1 log unit below. They're about 1 log unit below the wild type. They're all, we've lost a bit of activity, but you know, these are new variants with MET activity, experimental MET activity. And so finally, I want to talk now about a real application to try to search for beta-methionine activity. So beta amino acids, this is a schematic here. They have, of course, an extra methylene group in their backbone. And it can be either on the right of the alpha carbon. Here's the alpha carbon, the ammonium, the carboxylate, and the methylene can be on one side of the alpha carbon or the other. It turns out we've been targeting this particular variant. There are 2 classes of beta amino acids. We've been targeting this particular variant, including beta-methionine, but also a few others like beta-valine. So this is beta-methionine. Now, this is another non-standard backbone. And you know, this could be, again, to be able to introduce such an amino acid into the genetic code and open up, you know, put it into the toolbox of protein engineering, that could be really interesting, really useful. For example, you could have a beta sheet where instead of the side chains coming off, you know, alternately on one side or the other, you know, now they would come off all on the same side of the beta sheet, things like that. So this is not a theoretical proof of concept. This is really could be a very useful object for protein engineering. We stuck with 3 variable positions in the MET active site. We did a lot of other things as well, but the most interesting results were obtained with 3 variable positions in the active site. 200 variants were sampled. Remember, our sampling is exponentially enriched in tight binders. These are not random variants. These are the tight binders, okay? So 206 is not very large, but those are the ones that are exponentially enriched based on their transition state binding. 11 were tested among the top, I think it was the top 20. Our colleagues picked out 11. All of them were active. There was very reasonable agreement with experiment for the log catalytic efficiencies. Now the mean error is not half a kcal I showed earlier. It's more like 1.2 kcals. For catalytic efficiency, I'm personally, you know, really happy to have such an error level. But you know, that's my personal feeling. And what is nice is that 3 of the variants have a significantly degraded alpha MET preference or a significantly improved beta MET preference. Okay, we actually don't reach a preference for beta MET, but we reach variants that no longer lose their alpha MET preference. Okay, so the reduction factor we have 3 or 4 mutants with reduction factors between 10 to the 2 and 10 to the 3. And this is not yet published. Experiments are still ongoing, so I'm not going to show you full details, but these variants are already a nice level of success in going towards beta-methionine activity. Now, I spent a lot of time giving you background and methodology, and I'm not really going to have time for a whole lot of interpretation of these results. You know, why did this mutant work? Why did that mutant? Why was that mutant not as good? I focused on the methodology here and a few proofs of concept. I'm just going to finish by coming back to electrostatics. So as I said earlier, there are a lot of variations of this adaptive landscape flattening idea. There's one that is not on this list, and there are actually a bunch of others that are also not on this list, and that I'm not gonna describe here because we're working on them right now. But one that is not on the list has to do with acid-base reactions. Now, in the protein design field, acid-base properties are a classic benchmark for protein design models, right? Because protonation, deprotonation of a side chain looks a lot like a mutation. And the protein design software is perfectly happy to take, you know, protonation, deprotonation data instead of mutation data. It really doesn't know the difference. So this is a classic benchmark in the field, and we've done several rounds of acid-base benchmarks, and other people have done their own benchmarks, and we do reasonably well within this protein design subset. So this particular benchmark had 10 proteins, 149 experimental pKas. Here I'm showing you kind of a nice subset. It's the hemoglobin data, which is actually particularly nice. Again, the one unit errors are shown above and below the diagonal. And these are the overall statistics. Okay, so we have this physical model which has some elements of realism. It also has some drastic approximation, like a fixed backbone and discrete rotamers. And what we get is a 1 pH unit RMSD error, which is slightly poorer than ProPKA and slightly better than a null model. Going to the cases which have significant shifts, now we're doing as well as ProPKA. We don't really lose much, whereas ProPKA starts to degrade noticeably. We also have a small but significant improvement compared to a simplified GB model with an effective dielectric boundary. So the many-body GB here, this is another situation where the many-body GB gives a small but systematic improvement. So this benchmark was initially done as usual by doing pH scans and computing titration curves. But remember, if we view the protonation, deprotonation as mutations, we can also flatten the landscape in the sequence space with an adaptive bias. And that will give us another route to extract the pKa. So, this was actually pointed out to me by a student. I said to him, that's a good point, but, we've done a lot of pKa calculations. We have all this stuff going on. You should really not spend time on that now. You should try to finish up all these other design projects we have going on. pK benchmarks, maybe some other time. And then I went away from the lab. I left and I went to our house in Brittany for 2 weeks. And when I came back, he had done a complete study. So we actually published a paper. It's kind of a neat paper. The physical model is unchanged. And so indeed, we get back the same pKas within the noise level. The only thing that's changed is the sampling strategy. And the earlier sampling strategy was valid, the new sampling strategy is valid, and they both give the same results. It turns out the new strategy, the adaptive landscape flattening, gives us a factor of 2 to 3 in terms of speed. It's actually more efficient. And it's also very interesting conceptually. I mean, at least to me, it reminds me, I think some of the older folks in the audience remember the multi-canonical methods that were introduced in the 90s, where you were sort of simulating a whole bunch of temperatures at the same time, not at all through replicate exchange, but through a more subtle method. Here, we're effectively simply simulating a continuum of pH values at the same time. And we can back out the pKas through an unbiasing procedure. So that was my second electrostatic slide. And with that, I'm finishing, I think, right on time. I'm ashamed to say I don't have a photo of the student who really started out the adaptive flattening work, Francesco Villa. He and this other student, Vaiti Opu, who was here, these were shockingly good students, both of them. I've had a whole series of very good students and postdocs, but these two guys were really shocking. And Francesco implemented the many-body GB. He implemented the adaptive landscape flattening. He did a lot of testing and benchmarking. And then Vaiti not only did all the enzyme applications, but he also did the PDZ redesign. So that's pretty impressive. I've had some great external collaborators. The Many-body GB, I forgot to say, was work I did long ago with my very old and dear friend, George Akontas, the theoretician from the University of Cyprus. And then these are two experimental collaborators who have been really great, Ernie Fuentes at the University of Iowa and Yves Michulam here in Paris. And so, to finish, I'll just, say COVID fatigue is real. And, confined here in Paris, I think all of us are really tired of only seeing pictures of Paris. This is a picture I took myself last summer. We had this brief period of freedom, but I really look forward to seeing all you folks in the flesh. And so thank you all. And I think I'm right on time. Happy to take questions.
Thank you, Thomas. That was a really, really interesting presentation. Before people are starting, I see many people appreciate your presentation too. Before other questions arise, I have actually a couple of one myself. And the first is, how many mutation can your method afford?
Good question. Very good question. Thank you, Walter. We can adequately sample, adequately flatten a couple 100,000 variants, so if you have like full combinatorial freedom on, I think, 5 positions, gives you about 200,000 variants, and we can flatten that space. Or we can have a larger number of positions with this fewer allowed amino acid types. But 4 or 5 is huge, because we don't need to go directly attack 4 or 5 positions at once. We can do a systematic scan of pairs, designing pairs of positions, which is very cheap and easy. And picking out the high-performing pairs, we can group them to form, let's say, a quadruplet. Okay, and so if we do that, we're making the assumption that the correlations between the positions are pretty well captured at the pairwise level. So that's analogous to the Kirkwood superposition approximation. So if we select pair positions based on pairwise statistics, then we group them into, let's say, a quartet. Now we have the full correlations within the quartet, and we can go up to 4, 5 positions in a second phase like that. But in the first scanning phase, we can do 20, 30 positions, no problem. We can scan the whole active site very fast. A full study of an enzyme is a couple of days on a single desktop machine. And a billion Monte Carlo steps with the many-body GB is a few hours.
I see.
With 16 cores.
And the second question is, do you think, or have you ever envisioned applying a similar approach to protein-protein binding interfaces?
Sure. We've thought about it. We haven't gotten around to it. But yes, of course, the methods, many people who do protein design obviously have designed, there's some amazing applications from the Rosetta community. Of course, the methodologies that would be the same, yes. We don't have any examples or any successes there.
Okay, thank you. I have a question from Jean in the chat, which is saying, naive enzyme question, where greater variation in sampling, Km or Kcat?
Okay, that's an interesting question. We've done a lot of applications where we just sampled for Km. We targeted Km. Those were basically our substrate binding designs. I don't think we really tried to quantify. Certainly things were not noticeably harder moving to the catalytic efficiency. We can pretty well cover spaces of the same complexity, but unfortunately we haven't quantified, is it harder to, design, is it harder to design for catalytic efficiency or for binding in terms of the sampling effort? My feeling is it's very comparable.
Okay. Then there is a question from Wei.
Hi, Tom.
Hi, Wei.
Very nice talk. Really like really like your talk. Like to see it, the charm body doing well in all sort of topics, right? So here's a top, just a clarification. So when you do this sequence-based sampling, so when you change the sequence, do you do conformal sampling again, or you pre-calculate?
We do mixed moves. So a Monte Carlo move will either vary a rotamer at 1 or 2 positions, or it will vary, you could make a mutation at 1 position, or possibly 2 positions with a random rotamer selection. When we, yeah, you can, yeah, you have a lot of levers to kind of design the details of the sampling. But basically, when we sample mutations a lot less frequently. So when we do a mutation move, we're typically going to do about 10 pure rotamer moves before the next mutation move.
I see. So, okay, right. So in that regard, Landau has a little bit of a drawback, as we know. A little bit comment actually about history. It's very interesting because Landau was started earlier than metadynamics.
Right.
And so the difference at that time, actually, in early days was actually metadynamics used a constant Gaussian, but the Landau allow you to change the unit weight, right? You can change the unit weight. Slowly, the weight will become a constant in the end.
Right.
So in 2008, I combined Landau metadynamics, right, which allowed the metadynamics to change the height. So which was motivation for Parnello group to develop raw temper metadynamics.
Oh, okay, interesting.
Right.
Now,
Yeah,
I forgot to say, it was on one of my slides. I forgot to say that the schedule for developing the bias is exactly borrowed from well-tempered metadynamics.
Yeah, you just change the height, right?
Right.
Height is changing. So in this case, unit weight. So that's the issue for, right? If you pre-compute, when you do sequence-based sampling, if you pre-compute the energy, which will work very well. If you do couple sampling, sometimes actually you may run into an issue, which is actually when the unit height becomes so small, and then the modification you can put in is very, very tiny. At the same time, the confirmation sampling has not converged yet. You may have a little bit issue. I don't know whether you encounter an issue in your.
I actually have a student who's spending most of the time sort of, instead of doing what I want him to do, he's more interested in exploring the kind of the sampling properties of our procedure. So, eventually it's also very interesting. Yeah. So I have a very smart student who's, one of the things he's looking at is the properties of the sampling. Up till now, we've been very pragmatic. We have not done a lot of systematic analysis of, to try to really optimize the efficiency. We've been getting good results, but you've noticed we've kept our problems relatively small, 3, 4, 5 positions. And for those cases, the enzyme examples I showed, it was basically triplets of positions. And there we have really, really good sampling.
Right. So you have no problem in seeing the sequence-based random walk, right? In your Ecole, when you generate the weight, in the end, if you have no problem to see the sequence-based random walk, which means you're fine.
No, and what I again, in the interest of speed, I didn't give you a whole lot of details about our sampling. We have some very aggressive sampling strategies, more aggressive really than what I described. For example, we will typically flatten both the apostate and the holostate. And that allows us to pick out weaker binders than we would see if we only flattened the apostate. So we typically flatten everything that we encounter, basically.
Yeah, yeah. Anyway, so this is it's interesting, right? Because actually I thought about sequence-based sampling years ago, but I gave up because actually I was concerned about actually it's very hard to generate the sequence-based random walk when I couple with common change. But it's really nice to see actually you can
Well,
you
I
can use
should
it
credit,
for
there's one person, well, there are two now because we've started doing it as well. But the one person who's exploring sequence space with molecular dynamics is Charlie Brooks. And, you know, he's cannot handle as many, as large a mutation space as Monte Carlo. But it's a very interesting line. And we had a paper.
Yeah, actually that's the issue for Charlie's approach. The multi-state
Right.
is really because actually is we know very well this is we call hidden barrier problem, right? When you try to do sequence-based sampling, your conformational space, right? Conformational space coupling actually can be really hard to relax, right? And so in the end, run into the issue. I think that's one reason I Charlie see that problem too. Right?
Sure.
But anyway,
thanks, Wei.
Thank you for the talk. I enjoyed it.
I see a question from Marilyn. Do your result, yep, in the chat, do your results connected to a multiple sequence analysis on the same active site?
Yeah, good question. Are we recovering sort of amino acid types that are sampled naturally? Sometimes. Sometimes. Not so much, I would say. Not so much, but I can't give you a really good answer because it's not something I've gone back and looked at very recently. But we also kind of a flip answer is we don't actually look because we also don't really care, and we expect to. I mean, it's one thing if we're redesigning from methionine for the wild type substrate, but if you're redesigning for something different, and if the flip answer is we don't actually care that much. We care about how good is our physical model, and then what do the experiments say? Right? Those are the two things we care about.
Another question maybe that comes a little bit from Wei's is, do you, and maybe I missed it, are you doing backbone motions or just side chain motions?
The enzyme examples I told you are all side chain. Now we have we can also do we have a hybrid Monte Carlo method where we do backbone hops and then side chain relaxation. And we invented a hybrid Monte Carlo method that allows us to do that to maintain Boltzmann statistics. So if you do something like back rub simulation with Rosetta, it's very powerful, but it's a heuristic sampling. We actually can do Boltzmann sampling for, let's say, a dozen backbones. And the other thing I forgot to mention is that, you know, experimental systems obey Boltzmann statistics. That's a good reason to try to mimic them. But with Boltzmann statistics, we can actually compute free energies. That's why I can talk about, you know, catalytic efficiency or Km or pKa's, you know, because we really are able to back out free energies because we have a controlled Boltzmann sampling. So for multiple backbones, yes, we have a nice Monte Carlo method. It's a hybrid method that where you do a backbone hop and then you do sidechain relaxation, and everything I just mentioned is a single move, which then has to be tested, and that's the hard part.
And so you recompute all the side chain energies each time you do the backbone hop.
Yes, but it's very efficient because you're just changing one single site or two sites.
And is the acceptance for the backbone hops with sidechain relaxation reasonable?
It's reasonable enough. It's not something we've extensively optimized. We put all of our effort in establishing Boltzmann statistics. You know, getting a working protocol, and then we really did establish Boltzmann statistics, which is hard to do.
Yeah, no, I mean, we're also Monte Carlo
I know
centered.
you
So
work, of course.
I'm
yeah.
very, I'm very appreciative of all the difficulties and the power. It's
Sure,
great
I
to see
know.
more Monte
Yeah.
Carlo. I think it's a super powerful technique that's underutilized.
Well, it's
I
been a
think
lot
we
of
both
fun
do.
for me because I've had to learn all about Monte Carlo. And
Yeah.
that was.
No, it's so good. Thank you. It was,
Thanks, Marilyn.
great.
Okay. Are there any more questions? Yes, there is one from Antonio.
Right. Hi, Tom.
Hi, Antonio.
Nice talk. I have a question. Maybe it's a naive question related with the sampling in sequence space. I never thought much about it, but what you are doing is the reason why it has Boltzmann statistics is because you are doing some kind of grand canonical Monte Carlo. Is that so?
Yeah, you could view it as a. I mean, it's not really grand canonical because we're not bringing in new sequences. We have a pool in our statistical ensemble. We have a pool of, in principle, all sequences, and what we're constantly doing is picking sequences to unfold and others to refold.
Yeah.
Okay, so it's not really grand canonical. You would probably, I mean, I wouldn't really be comfortable formulating a grand canonical scheme where you bring in a whole protein. I prefer to unfold them and refold them. Now, of course, the pKa's, yes, that's a semi-grand canonical. That's a very standard, completely standard. Of course, you know all about that.
Yeah. Okay, because maybe I was thinking about it the wrong way, because I was thinking about it in the sense of, okay, you have a sequence, and then you are switching you are switching individual amino acids with the reservoir, but that's not the right way to think about it, right?
No. We're simply picking a sequence to unfold, and another one to it's a partner sequence to refold.
Okay.
And that defines the mutation. So this is never discussed in the protein design literature, as far as I can tell.
Yeah.
There's some very good theoreticians in the protein design community. People like Jeff Sabin, but it's not really discussed. So.
Okay. Because my thought was, if it was something like more of a grand canonical thing, then you would be your free energy will be something different, right? Would be some kind of grand canonical potential or something like that.
Yeah. No, here it's really very, very simple. It's just conformational space.
Yeah. Okay. Okay. Yeah. Thanks.
There are questions from Jim.
Sorry, very silly questions. Well, the first one's quite a detailed one. If you were looking to engineer an enzyme and you had access to directed evolution, which many of my colleagues do in Manchester, in the institute I work in, what would be how would you sell to them the collaboration?
Thank you, Jim. Good to see you. Thanks for that question, because it reminds me of another thing I forgot to say during my talk. What I forgot to say during my talk is that non-canonical backbones are not amenable so far to experimental directed evolution. So there are ways, you can think about ways to do directed evolution for non-canonical backbone substrates, but it's never been done. And it's the ideas to do it are pretty tricky. And as far as I know, they're not really out there. I mean, we have a grant proposal with some really excellent directed evolution experiment specialists, which has been rejected twice. But in principle, with the ideas we propose, we could enter a directed evolution phase. The requirement to enter, one of two main requirements to enter a directed evolution phase would be to get the beta-methionine activity to be at the same level as the alpha methionine activity. And we've achieved that now. And so now, in principle, we can improve our enzyme by experimental directed evolution. Of course, the difficulty is you have the selective pressure. We have ideas on how to do that, but it's not obvious at all how to apply selective pressure for a protein that contains a non-standard backbone element. You're welcome to think about it. But so the answer is that for backbone, for non-canonical, applying selective pressure for non-canonical backbones in a directed evolution context is currently not something that's done. Or certainly I don't know about it, and my excellent specialist colleagues don't know about it either.
Thanks,
I mean,
Tom.
that's our goal. That's our goal. Our goal is to enter into a refinement phase with directed evolution. Now we're ready for that.
Demonstrated that, and it's impressive. The link with experiment. My very silly question, Tom, is there's not many times we really get scientific excitement. Well, there are a few times we get scientific excitement in life, right? A few. And one of mine, unfortunately, was watching Jay Ponder with his big computer printouts in the mid-'80s in Fred Rich's lab. And over weeks, he was getting closer and closer to refining what sequences were compatible with BPTI fold. The early.
Right, sure.
And he ran, as I recall, sorry?
He was on my slide, you may have noticed.
Oh, yeah. As I recall, he ran into a stop at main chain movement, right? So I'm frozen 35 years ago, remembering it. So my question is, how, in the first part of your talk, when you're talking about sequence compatibility with fold, to what extent does main chain move and is required to move? How much does it move between different folds? It's a very simple question, sorry.
Right, thanks. So if we design a PDC domain with a single experimental backbone, our sequence entropy is way too low. Okay, we don't produce enough diversity because we're stuck on this single PDC backbone. If we go up, we've more or less randomly chosen 3, for kind of reasons of convenience. And with 3 PDC backbones, now we get back the same sequence entropy as the PFAM set of PDC domains. So you don't need a whole lot of backbone variation to get kind of a reasonable level of diversity. Is it the right diversity? Does it, have the right properties, the right amount of covariation and so on? That's a different question. But we can get a very decent level of diversity comparable to the natural PDC set with just a few backbones, which differ by, half an angstrom to an angstrom.
Thank you.
Okay.
Actually, can I ask, are you playing around with changing van der Waals, softening the van der Waals, or doing things like that? Does that help?
That's a good question. That's an important, kind of ingredient, technical detail of protein design. Most of the protein design energy functions have soft van der Waals. So we don't use that. We use a completely different idea that was proposed by Shoshana Vodak a long time ago for protein design, where for each residue pair, we do a little bit of relaxation. We do 10, 15 steps of energy minimization before we compute the interaction. And by the way, that's not quenched Monte Carlo. That's for building up our lookup table. So once we've done that, the energy function doesn't evolve. It's not quenched Monte Carlo. So yeah, it's a different method. It has some advantages and also some drawbacks.
Right.
Okay. I think there are no more questions. So let me thank again Thomas for his presentation of today. And we'll meet next week with Marilyn, and we will have a roundtable afterwards. Okay. Thank you very much, everybody.
Thanks, everybody.
Thanks, Walter.
Bye-bye.
Bye-bye.